magic of eukaryotes is that they have a mobile cell wall and a cytoskeleton that helps them move. These features allow eukaryotes to engulf prey to eat them. Bacteria cannot do this since they have a fixed cell wall. They can only absorb nutrients from other cells, but cannot take them inside their own cells. Engulfing things not only works for taking in food, it occasionally allows eukaryotes to neglect eating their prey and instead to turn them into organelles. That is, the eukaryote captures the biochemical machinery of the prokaryote, or potentially another eukaryote, in a bid to use their biochemistry for their own ends. The classic organelles are chloroplasts and mitochondria. Electron microscopy shows how similar the chloroplasts and mitochondria are to prokaryotes. For example, in green algae, the chloroplast has two membranes. These consist of the cell wall of the original cyanobacterium and the cell wall of the eukaryote that engulfed it. There are other reasons to think that organelles are actually engulfed prokaryotes. First, the details of photosynthesis in eukaryotes are the same as those in free-living cyanobacteria. We also notice that herbicides work on chloroplasts just as they would on a cyanobacterium. Furthermore, chloroplasts also contain DNA, RNA, and ribosomes. These are the remnants of the genetics and cellular machinery of the original free-living cyanobacteria. Now, that's not to say that there are no differences. For instance, the genetic complement of organelles is often reduced compared to that of the free-living cells because some genetic functions were transferred from the cyanobacterium to the host eukaryote. So the evidence for symbiosis between bacteria and eukaryotes is overwhelming. We might now wonder, well, why did it happen? What's in it for the cyanobacteria and what's in it for the host? For the eukaryote, they get sugar from photosynthesis of the cyanobacteria. Photosynthesis can be massively efficient, and algae just keep on churning out more sugar than they could possibly need by a long shot. As long as the sun shines and there's enough nutrients around, the photosynthetic machinery can be a sugar factory. In turn, the host cell gives CO2 and nutrients to the cyanobacterium. There's also secondary benefits, such as protection for the cyanobacteria by the bigger and more mobile eukaryotic cell. There are lots of other examples of this. Instead of talking about cyanobacterial symbionts, for example, we can see the same relationship between dinoflagellate symbionts and coral hosts. The dinoflagellates leak sugar to the coral, while the coral host provides nitrate, phosphate, and protection. Now, bear in mind, it's not always fully obvious who's in control here. We are used to thinking of the host as being in control, like being farmers that exploit the algae for energy. But the relationship could be the other way around, namely that the algae figured out how to infect the host to get all those goodies that it might have trouble obtaining on their own. Another possibility is that algae figured out how to avoid being eaten by turning off the "I want to eat you" cell signaling of the host. Over time, a facultative symbiosis, wherein the two partners can get on well enough without each other, becomes more tightly interconnected between the host and the algae. Chloroplasts, for instance, have less than 10% of their cyanobacterial DNA left. They've given nearly all their independent genetic operations over to the hosts. The chloroplast still works because the host gives it the proteins that it needs. Not only this, but the host cell has translocated a lot of the original cyanobacterium's DNA and incorporated it into the host's own genome. Mitochondria are pretty much the same story. Mitochondria are genetically and structurally similar to free-living marine prokaryotes. In this case, the host cell gives sugar to the mitochondria, which gives back ATP, the universal package of energy. So two fundamental metabolisms of eukaryotes, photosynthesis on one hand and respiration on the other, were stolen from bacteria. Our innovation as eukaryotes was figuring out how to move and grab things. And that ability allowed us, as eukaryotes, to snag a lot of basic metabolisms that had already been invented, perhaps billions of years earlier.